May I have your attention, please? We'd like to get the program started. I think everybody's just about finished with lunch, so we'll proceed. For me, this is a very special treat, having the opportunity to introduce one of my fellow colleagues at Baylor College of Medicine. Dr. Bert O'Malley is the Tom Thompson Distinguished Service Professor of Molecular and Cellular Biology at Baylor College of Medicine. He was the first to discover that hormones act by regulating messenger RNA production in target cells. He also discovered the existence of co-regulators, the co-activators and core-pressors that regulate gene transcription. The co-activators have turned out to be the long-sought master regulators of the mammalian gene function. He, in addition to his outstanding research and his dedication to education and training, on the side, he's trained over 250 scientists and published 650 papers. He holds 23 patents in the fields of gene regulation and molecular endocrinology, steroid receptor and coactivator action, and regulatory, cell regulatory proteomics and metabolism. Dr. O'Malley is the founding father of the field of molecular endocrinology, among many other things that he's done in his career. He's a member of the National Academy of Sciences and of the Institute of Medicine. He's received over 60 honors and awards for his work, including the National Medal of Science from the White House in 2008. I'm going to be very privileged to be on the stage with Bert this next week at graduation, and it turns out that both of us are receiving an award, but it's being in his presence that's the important thing for me, anyway, as you might imagine. But Bert has agreed to have a question and answer session at the end of his presentation, and he's going to talk to you about omics. Bert?
Thank you. It's a great pleasure to be here today. This is an area I have a great interest in, and this is a outstanding group of people, so it's my honor to talk to you. So Bobby asked me today to talk a little about omics, O-M-I-C-S, which basically would be biomics relative to biology and medicine. So what does that term mean? You see this term spread around a lot of places. Omics is a suffix for a word that means totality. So omics, when it's attached to other words, means a comprehensive look at or evaluation. So for genomics, it would be a comprehensive look at all the genes. For proteomics, it would be a comprehensive evaluation of all the proteins, or for metabolomics, of all the metabolic molecules. So it's pretty simple from that point of view. Now, what might you measure when you're thinking of the totality of human biology in the year 2013? Well, we're now in a position we can measure virtually all the major processes in the body. Of course, you know that you can all have your DNA sequenced now, And you read about DNA and genomics in the newspapers a lot now. So that certainly can be analyzed. You can also analyze other molecules. So DNA, you have 6 foot of DNA that you inherit that's packaged up into less than the head of a pin in the nucleus of your cell. And this 6 foot of DNA contains 2,000 genes. Those genes represent your capacity. Yes, it's what you are, but it's your capacity. The genes are inert. They're inactive by themselves. What brings them to life is other molecules in the cell. So a little enzyme decodes the DNA into RNA, and the RNA molecules go out of the nucleus to another part of the cell and direct the synthesis of proteins. So proteins is the major molecular endpoint from the gene, but the proteins then run all your metabolism. And it's really the metabolism, your metabolism, that gives us a snapshot of what you are right now. So yes, your DNA can be all sequenced, and we could tell one of you from another in the DNA, but we couldn't tell what's going on in your body by doing that. We could tell predispositions to diseases and if you have a genetic defect. But we must measure the other things. We must measure the proteins and the metabolism molecules, because that's what ends up in the physiology of your body. And that is absolutely unique. So each one of you have your own biomic picture. And the biomic picture changes from day to day, depending on what from the environment impinges upon your body. Okay? And certainly, you're all interested in a process that impinges a great deal on the body, and that's space travel. So I'll come back to a couple comments on space omics here towards the end. So you have the 20,000 genes make 100,000 RNAs, and those code for well over 1 million proteins in your body. And then that directs the synthesis of many metabolic products. And what is metabolism? I think you generally know. Metabolism would be the processing of all the foods you eat, sugars, fats, proteins. It would be the end result of all the hormones in your body and any chemicals that impinge on your body, environmental chemicals. Okay? So metabolism is an area that we could only measure a few things years ago. But in all these areas now, we can measure everything. We can measure all the proteins in a cell now. We can measure most all of the metabolites. And we used to have large amounts of material, blood or tissue to do this. Now, a couple hundred cells or very small samples, you're able to do this. So we're approaching now an area in biologic research that I think has an immense relationship to medicine and aspects of medicine that you're interested in. Why would you want to look at everything? Why not a few things? Well, because we don't know what few things to look at. You know, George Egel, in the early 19th century, coined the phrase, "the whole is greater than the sum of its parts," which I think you've all heard. And that's certainly true. If I laid out in front of you all the individual parts of an automobile, it would make no sense to you and it would be of no use to you. But once you assemble them into a functioning machine, it makes sense and it's of great use. So the assembly of the totality of information is powerful, very powerful, and we can do that better now because of computation and computers. So if you think of an iceberg as another example, if you're on a ship and you see an iceberg, you see this rising out of the water, and you're looking at 10% of the iceberg, just the tip of it. So as you go down deeper and deeper, you get to see the rest of the iceberg, the other 90%. So it's the same for biology. If you look at an individual, you see the pheromone, the so-called pheromone. And that's the picture that you get. And that is immediately related to the metabolism of the individual. And that's related to the proteins, to the RNAs, and back to the genes. So if you measure everything, you have a complete picture. And I'll tell you why it's important to do this and what are the advantages of that in a second. So to measure all these different things, how can we do this? What kind of samples, biologic samples, do we need? Well, for a mouse, you have no problem. You can look at all tissues, okay? You can look at every organ. You can look at blood, urine, everything, okay? For an individual, we choose not to be invasive to the body. Even a needle for blood is slightly invasive, and people don't like that. But there are other tissues that you can use. You can use urine. You can use saliva. You can use breath. Okay, how might this occur? So you might take an individual as you create a biome for an individual. And let's say one of the better parts of ways to do this is to take some urine samples over a period of time. Now, urine, in the earlier days of medicine, for some of you that are my age would realize that everything in medicine was measured in the urine many years ago because we didn't have the sensitive assays for blood. So the urine is a good source of material. It's a comprehensive source of material. You have 100 trillion cells in your body. These cells are constantly growing, dividing, or popping open and dying. They release materials from the cell. Those materials are a picture of what's going on in your cells, in your body. This material goes into the blood. It's in tiny amounts in the blood. And there's lots of other proteins in the blood that get in the way often of measuring. But the blood is homeostatic. That means what goes into the blood, if it's not meant to be there and doesn't have something to hold it there, where does it go? It goes out with the kidneys and it goes out into the urine. So the urine collects this material. And protein analysis you can do very nicely on urine now. Many of you might say, aware of medicine, well, people don't have protein in their urine. Because when you go to the doctor and you have a urinalysis, the urinalysis, if it's totally normal, it says no protein. Well, that's a laboratory analysis in the hospital. But if we take that urine sample and we go to high-resolution mass spectrometry analysis, we see now in the lab at Baylor 3,000 proteins in the urine. We have a good picture of the proteins in your body. The same holds for the metabolites, the metabolism end products. They come out in the urine too. So the urine is a good way to get a picture. Now, once you get a picture from the urine, a complete picture, you create a database of that individual or multiple individuals. And then you try to find, okay, now, this is the picture. And maybe with this person changes from this state to this state, a disease state, a living state, a state from Earth to space, many things will change. Then you say, well, maybe 500 things change. Well, we don't need to measure 500 because we will look in those and we will say, okay, these 4 out of the 500 always reflect the other 500. So we only have to measure 4 things. So therefore, we use now little handheld machines. It's essentially a cell phone that you can either spit on a disc or you can breathe into this little disc. And it's set up on an Android cell phone, actually, in the laboratories we're working with right now. That's a very cheap cell phone that you could get. The little chip does the analysis, and it sends it by telemetry back to a central reservoir. Just like now, when you have pain in your chest and you're picked up by an ambulance, in the ambulance, they put an EKG on you, like an electrocardiogram of your heart. Well, usually they just looked at it there. Now this goes back to the hospital. So by the time you get to the hospital, they know what's going on with your heart, and they're all ready for you. Well, the same thing can be handled with these analyses now. Just breathing into the machine or just spitting into the machine, if you have the database, can allow you to predict changes in state. So this gives you then a biomic fingerprint of that individual. And that is totally different in other individuals, because as 5 individuals move from one state of life to another, they will not respond the same. You can train them and train them and train them, but the bodies are set up differently. They will respond differently. It's nice to know how they will respond and whether they're responding correctly and whether they need some intervention, you to do something to prevent some untoward or deleterious effect. So what do you use these databases? Well, we use these databases now to look at child development, normal development, perhaps aging, normal aging individuals, and certainly in disease, because the omics evaluation can tell you something about the severity of a disease you have, how fast it will progress, and how you respond to therapy. So disease, yes. And also, one application could be space and space travel. And I'll give you one example of seeing something just going on in the lab now that relates to space travel. Of course, you all know that you have a circadian or a diurnal rhythm. There's a little part of your brain called the SCN nucleus, okay, that's sensitized to light. And you have a waking period of the day and a sleeping period. These have evolved in animals and humans for many hundreds of thousands of years. If you were a caveman 20,000 years ago, or woman, you would get up in the day and you would spend all day hunting for food, and at night you would sleep. And animals in Africa would still do the same thing, either in the daytime or nighttime, depending on what suits them. So when this cycle is disturbed, many things happen to the body. It sounds like a rather simple thing. Well, if you just disturb your sleep or your light-dark relationships, why should that matter so much? Well, it has immense relationships, and there's a whole medical literature about the associations of this with disease. But for space travel, it's about performance. If you go without sleep, things happen. So normally people need about 5 hours of sleep, okay? If you get less than 5 hours, you have a 20% decrease in performance. If you have less than 3 hours, it now jumps down again and can go as low as 50%. If you go 3 days without sufficient sleep, very poor sleep, you lose 70% of your performance capability. That's enormous. That means you're operating at 30%. Your decision making is 30%. Your physical function's 30%. Your mental function's 30%. So sleep is a major thing. So we're looking, say, we're doing this in mice now, sleep depriving them and changing their diurnal rhythm and looking at what happens to all the parts of the biomic pattern. This type of thing needs done for humans under certain circumstances, and space travel is a real opportunity, and it's something that should be considered. That you do this, and you might measure this over a period of time, first in mice, and then create a database, then check out some things in human. And eventually you should be able to get down where you can make some measurements by simply breathing into a small apparatus or spitting into it. It's all about collecting the information, though. And I think already in the space programs, many lost opportunities have occurred where deprivations or sleeplessness studies and so forth have gone on, that this information has not been collected. What a wonderful database if we would all do this and submit it to one database, and it's available to everybody. This would allow great predictive power for changes and allow you to do something. So, you know, what if we didn't know when you sweat on a hot day and exert, like a sports player, that you lose salt, and you need salt and some other minerals? Well, you know, on the football field now they give them Gatorade. It's a very simple water and mixture, flavored mixture with salts and some minerals and a couple of vitamins in. And this replaces the hydration. 1% dehydration starts affecting your decision making, 1%. And on a space shuttle, you may be operating with humidity of 20%, which is way below what you might see normally in life. And if you don't keep up with this, you start having changes in performance. And it could be up to the individual to drink something, but you know, they're not very good at this. An individual, you know, when they become dehydrated, they first, with very small dehydration, they become thirsty. After that, when the dehydration gets more severe, they're not thirsty anymore. So it's a dangerous situation, but these things can all be picked up by, again, biomic analysis. So I guess not to spend too much time talking today, the message is pretty simple, and I would say that it certainly is something that space medicine should be considering now. A giant database, which these new measurements that are capable now are made on maybe small animals like mice first, and then humans, and collect the data in such a way that we have predictive power, not for that you're going to get a disease, but for performance, for decision making, for mental function, for physical function. So I think I'll end there, and thank you. And if you have, I don't know if you want questions or not, I'm glad to do whatever you wish. Thank you.
Are there some questions? Dr. O'Malley.
Can you, is it possible to save the spit and the breath and the urine on location and analyze it weeks or months or years later?
Absolutely, yes. The answer is yes. Mm-hmm.
So if we're in some frontier
so the breath would be
environment.
saved by breathing into the machine or spitting on the detector, and that information would immediately come back to Earth and be saved automatically on the computer. For urine, I don't know what's possible to analyze on a spacecraft. You might analyze a few things, but you wouldn't be able to do probably a complete analysis. But the urine could be stored, samples, periodic samples just, brought back, and with a preservative in there, they could be analyzed. Because right now, even if something breaks down a little bit in the urine, we can measure the piece of it, and then we can fit it back into the whole protein, or the whole RNA, or the whole gene, or the whole metabolic pathway. So it's a we're really at a new state now in terms of biomic measurements, and so that's quite feasible.
This is Lakshmi Pucha from NASA. I think you answered part of my question, but I was just going to ask you, we have a repository of samples that we collected over the years from in space. So you think some of these analysis methods will be applicable for the stored samples?
The analysis we're doing them on stored samples now. So the answer is yes, we can do it. Because we will, for instance, if you want to talk about a mouse, we could collect urine over some weeks or months, and it's collected, and then we treat it in a certain way, and then you can analyze it at the end of that period. Sometimes you could desiccate it, that is, remove the water, it goes into a little desiccator, or other times you can add maybe a chemical that's inert.
Bill Pulaski from NASA. Just to let you know, we're already embarking on a program in this direction. Marshall Porterfield from NASA headquarters has started trying to pull together a large team to develop a NASA program to do omics analysis, probably starting with animal species and then moving on to humans in the near future.
Well, I congratulate you on moving to proteins. The only thing I would encourage you is, I don't know the level of your omics right now, but I now say we can measure every protein in the body. If I have a piece of your liver, I can measure all 14,000 proteins in that liver. Or if I have a cell of yours, I can measure in one cell all 7,000 to 9,000 proteins. So I just encourage you to take advantage of the totality of the measurements possible. Don't pick and choose what you want to measure by what you know today, because we have a lot of new things that are going to come to pass and that we're going to find out that will be predictive for the future.
Well, the idea is to look at the whole spectrum of omics and to make an available database that people can...
That's exactly what I'm saying needs done. You need to have an omics database that hopefully every country interesting, or maybe even commercial flights for the future can use, because this is serious business, and anything we can do to better prepare or to prevent unexpected happenings is going to be better. And these are, compared to all that has to be done to send spacecrafts in space and train people for years as a cosmonaut, these analyses are a small add-on if you do them right and comprehensively.
Yeah, the idea is to have a large database that would capture all of this stuff and be available to researchers and other space agencies as needed, and probably for research proposals against it for things that we don't even know today.
Good. Well, that's good.
Are there things we should be, Kate Rubins from NASA, are there things we should be thinking about for sample collection technologies? It seems like the point of care analyses are great, but it takes a lot of work to design and fly hardware. So if there's ways that you can collect, fibroblast samples from the skin or concentrate urine, that the sample collection and correct preservation and to then ship back to Earth to do the complex mass spec analysis is absolutely critical, that you're collecting the right samples at the right time points in the right way.
Well, there's been a couple of questions on collection, and I say you're right on in that collection is a large part of it, because if the collection's faulty or incomplete, or you're collecting the wrong things, you can have the best machinery and scientists in the world, they won't get the complete answer. So we're after biomic analysis. We don't want a sample to come in that we can look at and we can determine 30% of what's in there. We want the 100% now, because we know there are a lot of things that never could be measured before that are going to show up, that are going to have much greater predictive power for the future than the things we know about right now. We're pretty good problem solvers, so if we know sort of what to collect and how much, I think I have confidence that a combination of the biologists, the physicians, and certainly the engineers will help us get that material. Okay. Oh, thank you.
Piece of this is the phenotype, which is to know what the individual was capable of cognitively, or if you're looking at bone loss or muscle loss. And we have to do a very good job with assays for phenotype or establishing phenotype. And that means a robust documentation of interclass correlation, high variance that's stable and trait-like over time, so that we can line up that terrific power database with what the real phenotype is of the individual. And I think that that's the phenotype part is sometimes obvious. It's, a clinical test gives it to you, but sometimes you have to repeat exposure against a state variable to get it. And so the only other caveat here is that may mean you do some ground-based work on astronauts before they go up, looking for differential responding or stability of phenotype to cold exposure or sleep loss or other things.
Yes, that's excellent point. It's, I totally agree. I mentioned the phenome, but I didn't spend much time on it right now, but more the analytical technologies, but actually that's just as important. So you're really looking at the phenome, metabolome, proteome, transcriptome, and the genome, you know, all that stuff together.